never get my part in the center. Well, today's lecture is the continuation of the detailed study of the hypothalamic pituitary gonadal axis. And I'll switch to the computer. In this lecture, the lecture D, we'll be discussing the Gnrh receptor, or Gnrhr, the gene and its promoter region. And then we'll go on to talk a little bit about the release mediated through this receptor of the gonadotropins. And then lecture E will focus on the gonadotropins, the promoter regions. So an important thing to note is that for nearly every gene that we've studied so far, or hormone or receptor, we've looked at the gene itself, the exons and introns. For the LH and FSH, we will not evaluate the genes themselves, how many exons and introns or things like that, but we do focus on the regulation of the gene expression, and then, of course, at the end of this lecture, the release. So let's begin to talk about the Gnrh receptor. The notebook is on the button. You're on television now. Forever recording. It is a membrane receptor, as you recall. We are considering it on the gonadotropes, although there is some expression of the receptor within the brain for extrahypothalamic circuits. Our focus is just on the gonadotropes. And it is, as you recall, a 7-membrane transmembrane spanning class of the receptors, G-coupled, as you recall. So 7 transmembrane spanning segments. So immediately this should be telling you that when we talk about the genes, the exons will contain particular transmembrane spanning portions of the receptor, and extracellular loops and intracellular loops. So that's kind of a challenge. This receptor is interesting in that it does not have a cytoplasmic tail. So the classic model that we had studied showed 3 domains: the extracellular, the transmembrane, and the intracellular. But that's missing for this receptor. This would tell us then, of course, that the G-coupling portion of the receptor must be either on one of the intracellular loops or maybe the carboxy terminal tail that is certainly on the cytoplasmic side. Just doesn't extend in as we had seen on other membrane proteins and receptors. So it is coupled to adenylate cyclase, as we had stated. The G sub S is the case, not the only potential coupling mechanism that we're going to look at, but it is a definite one that you would expect for any of the hypothalamic releasing hormones. And of course, the cyclic AMP is the second messenger system, and the question is whether the G sub PLC or phospholipase C enzyme system that produces the IP3 and the diacylglycerol is involved in any of the mechanisms by which the receptor produces its intracellular effects. Certainly it triggers synthesis and release of the gonadotropins. We state that all the time. We expect it. And the question we ask, of course, is how. So trying to build a bit on our critical thinking at this level, of course, and interpretation of the scientific literature. So here's the receptor. There's the extracellular domain, 35 residues. It's the amino terminal. You see these bolded letters are sites of N-glycosylation. So we've stated that the extracellular tail typically will be glycosylated for receptors or way back. Transmembrane spanning or integral proteins are typically glycosylated extracellularly as they function as receptors. And those glycosylated residues, the carbohydrates attached to them often are involved in the specificity of the ligand, of the receptor for its ligand, I guess we should say. There's a transmembrane spanning portion as well as the loops. So remember, with the 7 transmembrane spanning segments, you're going to have intracellular loops and extracellular loops. So somewhere within here are the lipophilic domains for the transmembrane spanning segment, and then the hydrophilic domains that tolerate intracellular cytoplasmic and extracellular interstitial aqueous media, as we might predict. And so you see that there is no third domain shown here. Back up just a moment. Notice that there are 2 sites of potential N-glycosylation. One was the N-terminal extracellular domain, and the other was in the first exoplasmic, or we call them cytoplasmic or extracellular loops. So this is the first portion of the extracellular loop in here, a portion of the first extracellular loop in that region there. The regulation of the receptor, in essence, we're thinking at the level of gene expression. Estrogen, estradiol, increases the number of Gnrh receptors. Remember that this is part of the estrogen priming, if you will, in anticipation of the LH surge. It will sensitize the gonadotropes to Gnrh, and that is through the increased expression, or increasing the number of the Gnrh receptors expressed on the gonadotropes. And then ultimately we know the effects on the Gnrh pulsatility, the magnitude and the frequency of the pulses. Progesterone decreases the number of receptors. So this has been demonstrated in vitro only at the time, about 2000, I guess, or maybe 1999, of searching the literature. So this may, there might be more known about that at the time you view this lecture. But what was demonstrated at that time was that there would be about 500 Gnrh receptors per gonadotrope. Again, this is in vitro, so they can get the cell number. And that's under the progesterone influence. So obviously that's a component of negative feedback. You desensitize or you downregulate. If we're talking about this level of receptor reduction, you have downregulated. Remember, we said for downregulation there was not only internalization and probably destruction of the existing receptors, but decreased expression of the receptor gene. So certainly predictive involvement or role in negative feedback mediated by progesterone at the level of the gonadotropes in the anterior pituitary. And then the estrogen contributing its positive feedback effects, increasing that to about 20,000 receptors per gonadotrope. And this is also in the presence of inhibin. So both of them together will also sustain this level. So they don't potentiate or they don't have an additive effect. Either of them can, or both of them together can bring the number of receptors per gonadotrope up to this level. Again, because they're talking about the number of gonadotrope receptors, most likely they're performing some type of radioligand binding assay that does allow you to estimate the number of receptors, and because it's in vitro, they have the number of cells, so that total number of receptors divided by the number of cells gives them this estimate. Whoa. Then we can begin to think about the regulation of this gene and its transcription. So there was a hypothesis that during luteolysis, remember luteinization is when the ovulated ovum turns into the corpus luteum. Luteolysis is the other end of the spectrum, the death, the lysis of the corpus luteum. So at that point, of course, you are seeing the apoptotic kind of a programmed cell death of the corpus luteal cells. So obviously the synthesis of progesterone and secretion is going to decrease significantly, as do the plasma levels. As we know, at the end of the luteal phase, this becomes quite dramatic. And this will lead to an increase in the GnRH mRNA and the number of receptors. So this was the prediction at the time of the article from which I've extracted this information, very likely in the late 1990s. So the test was that on day 11 or 12 of the estrous cycle, they would induce luteolysis with PGF2 alpha. So this will, this hormone automatically, when it's given exogenously, will trigger the apoptosis and luteolysis. So within about 12 hours, there's this significant reduction of the progesterone, and of course the outcome is that when you measure the messenger RNA in the anterior pituitary for the GnRH receptor, it will have increased about twofold. So that's what you'd predict if you just eliminated hormone 3 that was involved in the negative feedback. You would expect an increase in the gene expression of the receptor at the level of hormone 2. Hormone 1's receptor on the cell that synthesizes and secretes hormone 2. There is no change in the number of receptors, however. So you have more message, but if it's being translated, it is not producing receptors that are present in the membrane. So most likely, we would begin to think that this would be a stable messenger RNA, or there's an increased turnover in the number of receptors, so that as you're putting new ones in at a higher rate to match this mRNA increase, they're being removed nearly as fast and eliminated. So a couple different ways of thinking about that. So this type of a study creates just an association between the progesterone and the GnRH receptor gene expression. So there was no evidence of direct effect of progesterone on the expression in this case. Of course, we know you have to turn to the promoter and look for progesterone receptor, progesterone response element, by sequencing, all those things for promoter region analysis that we had talked about, as well as using the functional assays, the transfections with the reporter gene, the approach that we've seen so many times. As it turns out, there's only a single copy of the gene, and in the humans, that gene is on chromosome 4. It has 3 exons and 2 introns. So already we should be able to picture in our mind that if we've got 7 transmembrane spanning segments and the intracellular and extracellular loops, this is a long protein, so these are probably going to be big exons. Exon 1, of course, 5' untranslated region. However, in this case, it also includes transmembrane spanning segments 1, 2, and 3. So already in your mind you should picture the N-terminal through transmembrane spanning segment 1. So you'd have the first intracellular loop, TM2, then the first extracellular loop, TM3, then the third intracellular loop, and we move into the fourth transmembrane spanning segment. So from that analysis, that vision in your mind, you should be able to determine and make a list of the extracellular and intracellular loops that are contained within exon 1. Then exon 2 continues through the remainder of exon 4, transmembrane spanning segment 4, and then, where were we? In, out, in, and then got into 4, so we do the next out, extracellular loop, and then we have transmembrane spanning segment 5, and we get back in. So part of the third intracellular loop is at the tail end of that, and we pick it up with exon 3 for the final portion of the third intracellular loop, go back out through transmembrane spanning segment 6, complete the final extracellular loop, and then the carboxy tail is at the end of the 7th transmembrane spanning segment, just kind of poking its nose into the cytoplasmic side. And then, of course, our expected component for the final exon, the 3' untranslated region. So that gene is shown in this slide, as well as its promoter region. So it provides everything that we typically discuss if we have our hands on the information. This is coming from a 1997 publication in Molecular and Cellular Endocrinology, volume 130, issue 1 and 2. And it is showing the schematic representation of the rat Gnrh receptor with exons 1, 2, and 3. The transmembrane, of course, the 5' untranslated region is shown, and the 3' untranslated region with the poly A tail, the potential sites for the polyadenylation, for the addition of the poly A tail on the 3rd exon. And, of course, the transmembrane spanning segments, first part of 4 at the end of 1, second part of 4, and then 5, and then 6 and 7 on exon 3. We're being reminded here that the primary transcript, the initial complete transcript is processed, that RNA is processed, and actually there's 3 different sizes, a 4th will be mentioned later, of transcripts, basically messenger RNA transcripts that can be isolated. 4.5 kilobase, 3.5 kilobase, and I think that's a 1.6 kilobase. So this immediately tells us if we have one gene and multiple transcripts, that is multiple isoforms of the molecule, then there must be alternative splicing that occurs. And the promoter region, got my pip on the promoter region there. On the promoter region, we notice an SF1. That's not unfamiliar to us. We know that that was essential. There's a couple of SF1s actually that's been essential for expression, the tissue-specific expression in the pituitary. Way on out, we've got the PIT1 for pituitary tissue-specific expression. And we see the CRE, so that's predicted with the adenylate cyclase, cyclic AMP, through the pKa, phosphorylating Krebs, and then the AP1 for phos-Jun sites. That immediate early gene is quite often involved in regulating the expression of many different types of genes, as we have seen. In addition, there's note that there's a CATAA that indicates a possible Tata box, and then out here a CCAT that marks the location of 2 possible CCAT factor binding sites, and we'll see the significance of that in a moment. And then the sizes of the mRNA transcripts were measured by northern blots, as shown over here. So remember, the northern blot, they isolate the message and probe with cDNAs, and there is an electrophoretic separation of the RNA, so you would end up with multiple bands, and you'd use standards to know what the length of the transcripts would correspond to. So that's how this evidence was attained. And when you see multiple transcripts from one gene, then of course, as we said, you conclude or you hypothesize, you predict that there is there is alternative splicing occurring. Well, this is the entire sequence of the 5' upstream portion out to looks like -1782, I guess, out there. And we see the PIT1 is shown here, the CCAT, another CCAT, the AP1, as we are, of course, snaking our way down through this. On the far left, there's -222, and here's -162, so it's an index of where you are along the length. We see here, if we go back to the -222, Here's the CRE. The SF1 is here. We saw the AP1, so those were the sites. Consensus sequences, if we're looking at the sequence and we recognize a particular consensus sequence, then we've identified them as candidate regulatory elements. You have to go on to the functional test to demonstrate that they are, that they can, first can they bind the cognate transcription factors, and then on to functional tests. Then even closer to the start site, when we see the capital letters appear right about here, that's for the coding region. So the second SF1 was really close to that. So these are the coding regions, and then we begin to see, of course, the predicted amino acid sequence that would accompany them as they're going along. This slide is showing us a comparison of the rat, mouse, and human, and there isn't much to comment on other than that rat and mouse promoter regions, as they had been sequenced or analyzed at that time, were quite similar, as we had just discussed. The SF1, TATA, SF1, AP1, the two AAT sites, the PIT1. They're just not showing the AP1 on there, I guess. And the 3 exons, just slight differences in the length of the 3' untranslated region. Nothing major for us to get all excited about until we get to the human promoter region, and we see that there's something going on there as well. The exon 1 is quite a bit larger, and of course, as we said, the exon 3 for the 3' untranslated region, but the 5' untranslated region is much longer. There's a lot more DNA here. We begin to see a TATA, a GRE, a TATA. I don't know what ENG is. There's more of these things than the brain can keep track of, I think. PIT1, the 2 CCAATs, a TATA, AP1, a CCAAT. I don't know what a PEA-3 is, but the list goes on. And then finally a CRE. So we recognize maybe 50, 60% of the ones listed on there. So obviously there's potential for a greater diversity of biological variations, if you will, if that'd be the appropriate term, for the human gene. So let's consider the human Gnrh receptor gene a little bit. We're showing it here. First, we go to the promoter region. We see that there's a progesterone response element that has been identified there as well. I don't think that was on the previous one. The glucocorticoid response element. So the PRE had, from this source, or the sources subsequent to the other study we quoted, the PRE had been found on the human Gnrh receptor gene, so there's that site for negative feedback through progesterone. Glucocorticoid response element, so the interaction of stress and the HPG axis. We know that typically stress suppresses the HPG axis, so we'd predict this GRE as a repressor or decreases gene expression. There's our friend PIT1 for the tissue-specific gene expression. AP1 for the CFOS, C-Jun family. The TRE for the thyroid hormone receptor. CRE, and then 2 more TREs. So the human Gnrh receptor gene, much more complex than what we had seen for the mouse and the rat. 18 transcriptional start sites, just seems bizarre. 7 TATA boxes, as we said, 6 CAT boxes, and the closest transcriptional start site is at the 577 bases from the 0 region. The 4 TATA boxes are present in the mouse, so still some potential for complexity of the different types of transcriptions. Whenever we see additional TATA boxes, these are alternate sites to begin transcription and produce a diversity of the primary transcripts that can be processed post-transcriptionally through different means. So these are 62 base pair upstream of the translational start site. So and note this is a translational start site in this case. So there's an additional 62 bases upstream of there, and it's uncertain what they might be doing. So as we've mentioned in some of the other lectures, from these types of molecular studies, information is extracted, and we might be able to make predictions, but it just doesn't fit the completely expected picture. Biology is more complex than what we initially predict, and molecular biological techniques certainly have added to that intrigue. I don't know what we're going to find. Hopefully we find what we predict, and who knows what else we might find, and that just takes us down other avenues to find out more complexities relative to the system. So this information explains the multiple transcripts, the 5.6, 3.8, 2.1, 1.3, and there's also been a 0.8 kilobase pair transcript that has been identified. At the time of gathering this information, I wasn't certain what all of those isoforms, or wasn't certain if each of those transcripts is translated, and if there is a corresponding gene product, the isoforms, and what biological function they might have. Now let's think about how GnRH might affect its own gene expression. So it regulates the mRNA levels of its own receptor. This is receptor regulation by the cognate ligand, so it's homospecific receptor regulation. So to investigate this, the GGH3 cell line was used. These stably express the GnRH receptor. And you can use these to test the response to GnRH and cyclic AMP to evaluate the effects on the expression of the GnRH receptor promoter. In this case, it was the luciferase reporter gene, and they used a 1,226 base pair portion of the promoter out to the plus 62 point. And obviously we're looking at transient transfections with this construct into the GGH3 cells. So we know that there is a stable expression of the receptor gene in these cells. So if we transfect with the GnRH receptor promoter and the reporter gene, we should have some baseline expression, and then we can play with the system, stimulating the receptor that is produced, and add some candidate second messengers to see what effect there might be. We already know there's a CRE, so we expect the cyclic AMP to have an effect. But what we're asking is the homospecific regulation, receptor regulation through GnRH mediated through the cyclic AMP second messenger system. So if GnRH is added, or an agonist called buserelin, then there is an increase in the luciferase, and it is dose dependent. This is gradual from about 1.5 to 6 hours, and it peaks at about 6 hours, and this would be a case where you would add the agonists and then wash them off. They'd be there for a certain period of time, I'm not sure exactly how long, and they're washed off, and then you measure the effect, the enduring effect on the expression of your reporter gene. So after that washout, there's a reversal back to a decrease back to the basal level at about 12 to 24 hours. So this is quite typical of a metabotropic, it's been so long since I used that term, a metabotropic type of receptor-mediated effect on gene expression, and certainly for a transiently transfected reporter gene system, such as this. We'll begin to talk more about cholera toxin as we proceed. It is a direct activator of adenylate cyclase, as we recall from a prior lecture. And then this DBC-AMP, it is a dibutyryl cyclic AMP. It's a synthetic analog of cyclic AMP, has slightly a longer half-life, so it can be used to test if cyclic AMP is involved. Freely diffuses into the cells as well. So either of these two also increase the luciferase. So again, we already know there's a CRE. We're beginning to see that even though we didn't have an article that reported binding of activated CREB to the promoter for this receptor, we're getting here indirect evidence that would predict that CREB is involved. When either of these two were added with buserelin, there was an enhanced luciferase increase. So they were additive, at least additive types of effects. So if these two, you're bypassing the receptor, going straight to adenylate cyclase or straight to the cyclic AMP mediated effects. If you combine those with the buserelin, then you can get an increase. So it implicates that the receptor is working through these similar systems. With Pertussis toxin that activates the PLC, our other standard candidate second messenger system, the PLC with the IP3 and the DAG, there was no effect on the expression of the reporter. Some of the concluding statements then, buserelin for 6 hours would increase the cyclic AMP. Adenylate cyclase inhibitors would have no effect on the basal expression of the reporter gene. And the also reduced the agonist-mediated luciferase increase. So we're not only going straight to the adenylate cyclase and activating it, but we're also inhibiting it and showing that there's no effect on basal expression, but there is an effect on the receptor-mediated increase. It attenuates that or prevents that. Using progressive 5' deletional analysis, these basically all this evidence implicating cyclic AMP mediated effect was retained up to -331, and once you truncated below closer to 0 with reference to the -331, then you began to get a decreased expression and responsiveness to the cyclic AMP tests that we've talked about. And it was identified that there's two CREs between the -331 and the -255, and the one at that level, and the second at -255 to the +62. Now, what you may find is as you're if you're beginning to pay attention to some of these various studies, they sometimes identify these response elements at similar locations, and then other times they kind of miss. So it's not always exactly the same from study to study. But typically it's quite reliable. Well, then we ask, what about in vivo? So great, you've got this permanent cell line that expresses the Gnrh receptor, and it works there. The next level is whether or not this promoter behaves the same way in vivo. So in this particular study, the ovine Gnrh receptor promoter was used to produce a transgenic mouse. Then you've got a whole animal to study the promoter behavior in vivo. So this allows you to investigate in vivo, but it's very challenging to do the truncated studies because it's much more work to produce a transgenic animal than to do the transient transfections in vitro. So in this case, they use 1900 base pairs of the ovine GnRH promoter, again with the luciferase reporter gene. Now, in vivo, this works well. There's newer reporter genes, a tremendous array of them, where the gene product fluoresces itself when it is excited. So you don't have to use the luciferase and provide a substrate and so on and so forth. So there are better reporter genes for doing the in vivo transfections, such as green fluorescing protein, and now red and blue and a variety of other colors of fluorescing proteins. So this 1900 base pairs does direct the in vivo expression, so we can get tissue specific, and we wonder then about cell specific gene expression. So 10 tissues were screened, and it was expressed in the pituitary, the brain, and the testes, as would be predicted. At least now that we know that there's basically almost a complete HPG axis in the gonads themselves. So continuing with this, you measure the luciferase in the pituitary, and in intact males and overreactomized females, treat with an anti-GnRH serum or with an agonist to see the responsiveness of your reporter gene, and then treat with an anti-GnRH and either testosterone or estradiol to see the responsiveness. So there is not, at least at the time of this study, I'm not certain if there is even today, a very good or any GnRH antagonist. So you can add an antibody that will bind to all the GnRH or enough of it and prevent it from binding to the receptor. So you remove hormone 1, expression should increase, right? And if you add an agonist, then you should be able to control the level of expression. Excuse me, did I say removing hormone 1 would increase synthesis and secretion, or the expression? It should be decreased, right? If it plays any role in the basal expression. And then treat with an anti-GnRH, again, to remove hormone 1, remove the receptor drive for it, or the ligand, the hormone 1 drive for the expression of reporter gene, and then test the effect of testosterone hormones 3 on the system for any potential negative or positive feedback. As it turns out, the steroids had no effect on the transgene. Remember, on the sequences that we showed, there was no ARE, androgen response element, or ERE on the promoter region, as it had been reported. So that was consistent. Using the anti-GnRH antibody, it decreased the serum LH concentration, as you'd predict. You remove hormone 1, hormone 2 levels should decrease, and hormone 3 levels should decrease. The pituitary luciferase also was decreased, If you added an agonist, of course, there would be no change, depending upon the concentration or the dose of the agonist that you would add. So what they're showing here is that the decrease that they're producing with the antibody is not altering the responsiveness of the receptor. It's removing hormone 1 and preventing it from binding to it. So how might we explain this? It is the target of this investigation, the GnRH, and how does GnRH cause the release of LH? So of course, the target of the antiserum is the GnRH, and we see that we can remove it and manipulate the receptor and still get the changes in the predicted direction. But how does the GnRH cause the release of the LH? Well, we recall that the gonadotropins are glycoproteins. They're heterodimeric. There's the alpha that's common to all of these, and the beta subunit that's unique for each. So for LH release, it is a G protein activation of the gonadotropes that's responsible for it. Obviously, this G protein activation will increase LH release, as we would predict. There's also an increase in IP, the IP3 inositol triphosphate production. So this implicates the G sub PLC system in this G protein activated LH release. There's a decreased responsiveness to GnRH. So we begin to think that as there's an increase in release, you will get a desensitization or maybe a downregulation of GnRH receptors as part of the stabilization or maintaining homeostasis. There's an increased responsiveness to calcium ionophore. This will be a substance that will increase the extracellular calcium influx into the cytoplasm. We know that when that happens, we should be able to predict increased exocytosis. So if you activate G proteins in the gonadotrope, increase the amount of calcium going in, then you will have an increased exocytosis. So this is implicating that calcium is involved in this G protein dependent exocytotic release of LH. And a decreased GnRH receptor binding. So this decreased responsiveness and decreased receptor binding go hand in hand for this downregulation. So this type of information is consistent with what might be happening after the LH surge, is that if you have times when there's elevated release of LH, even in males as well, then there's some mechanism within the gonadotropes to decrease the receptor drive to bring the levels of release back down to normal. Now, how that might occur independent of the effects on the LH alpha and beta gene expression, so that you can bring your synthesis levels back up to replace what was released, is uncertain from that discussion. But at the moment, we're trying to think of how GnRH binding to its receptor is bringing about release. The next lecture covers the synthesis to some degree at least. So next we might ask what particular G proteins. So if we activate G proteins, we get all that stuff happening, and we ask ourselves, how would we determine which particular G proteins? Well, this is where these toxins that cause ADP ribosylation of the alpha subunit of the G proteins become useful. There's the Pertussis Toxin that acts at a cis residue, and the Cholera Toxin that acts at an Gnrh residue on the alpha subunit. So might it be the G sub S or the G sub PLC? And these two tools allow us to differentiate. The cholera toxin is specific for adenylate cyclase, and the pertussis toxin is targeting the G sub PLC system. So the cholera toxin stimulates the production of the cyclic AMP, therefore. That's mimicked by that butyryl 2 cyclic AMP, the dibutyryl cyclic AMP, as we had mentioned. Increases adenylate cyclase activity. I believe I may have said that it acts like cyclic AMP. Corrected here that it actually acts on the adenylate cyclase and can increase its activity. The cholera toxin might have no effect alone, but it did enhance the GnRH, or this sodium fluoride and this calcium ionophore, stimulated LH release. So this means that the cholera toxin alone, activation of the cyclic AMP alone, did not have a significant enough effect to induce LH release. But it did enhance these other means of stimulating the LH release. So there's no effect on the GnRH or the sodium fluoride IP production, however. So what we're seeing is that there's some interaction between increased adenylate cyclase activity and the GnRH and calcium-dependent, extracellular calcium-dependent release of the LH, but it seems to be independent of the IP production by the GnRH or this sodium fluoride. The cholera toxin increases the percent of the cellular LH released to continuous or pulsatile GnRH. So it appears that there is some role for the adenylate cyclase in the receptor-mediated release of the LH. So there was no change in the total, the intra and extracellular LH, in this particular in vitro study. So this would make us think that there's something happening intracellularly to keep the intracellular levels at a high enough concentration so that we can sustain an increased extracellular concentration. So this type of a phenomenon would be consistent with being able to produce an LH surge. You have to have enough releasable in the cell to continue to be released to sustain the extracellular, remember, and that's ultimately going to be reflected in the plasma levels of LH. So this is part of that phenomenon that we're seeing. It's the cholera toxin does play a role, so it is G sub S dependent by some means. And this ability to sustain the intra and extracellular concentrations of the LH was thought to be from a redistribution from a non-releasable to a releasable pool of LH. So we're beginning to see these ideas in this earlier literature of how do we get the gonatotrope prepared to produce and sustain the LH surge and then be reversed, as we saw in the previous slide, to normal baseline levels after the surge and behave predictably in a negative feedback fashion. This ability is lost with homeospecific desensitization to high pulse frequency, about 1 every 15 minutes, or to a constant low concentration of GnRH. So these are parameters of GnRH bombardment of gonadotropes that do not match the endogenous events associated with the LH surge. But this balance between the intra and extracellular LH was restored by sensitization. In other words, sensitization was recovered by the increased concentration of the GnRH, or resetting the pulse frequency to 1 every 90 minutes for this in vitro test. So this investigation with the cholera toxin is implicating the adenylate cyclase cyclic AMP mechanism in this behavior of gonadotropes consistent with producing and sustaining the LH surge. So this pulsatile GnRH in vivo then, what is that? Well, there's interpulsive intervals of about 30 to 70 minutes. So we saw the 1 per 90 minutes in the prior slide. And the peak decays after about 10 to 24 minutes in the rats. So the 1 every 90 minutes was almost matching what in vitro in the prior study, what happens in vivo. So this is the pulsatility that is consistent with bringing about the LH surge. So the variation of the pulse frequency and the amplitudes, as we know, will differentially regulate the secretion and the biosynthesis of the gonadotropins. So it's scanty data, as you saw from the studies that we reviewed, but the data interpretation is consistent with what we would predict would be happening. Well then, what about the Pertussis toxin? Is there any role for the G sub PLC? The Pertussis toxin will when combined with the GnRH and the sodium fluoride, brought about a decrease in the IP production. Alone, there was no effect on the LH release, and the Pertussis toxin decreased the gonadotropin releasing hormone receptor binding, as of course do the other two. So it did produce the downregulation. Didn't seem to have any effect on release, which is interesting because the IP3 causes an increase in the intracellular release of calcium from intracellular stores and can also contribute to exocytotic events. So these data between the cholera toxin and the Pertussis toxin are implicating the cyclic AMP in the increase in extracellular calcium from extracellular stores. So taking another step in looking at the evidence for multiple guanyl nucleotide binding proteins in the GnRH action in this 1993 endocrinology paper, as we saw, the GnRH stimulates the IP production but not LH release through a Pertussis toxin sensitive G protein. A distinct cholera toxin sensitive G protein appears to provoke gonadotrope sensitization by stimulating an increase in the intracellular cyclic AMP levels. And there appears to be a distinct G protein insensitive to PTX or CTX that's capable of mediating the LH release independent of the IP production and the cyclic AMP. So there seems to be this third mechanism involved. So what we were able to substantiate from the review of those studies is that the GnRH receptor, when ligated, is coupled to G proteins, at least the G sub S, cholera toxin, studies showed that, that is involved in activation of the adenylate cyclase, and then ATP is converted to the cyclic AMP, as recall. It can target membrane protein kinases, causing ATP to be converted to ADP with phosphorylation of that and the separation of the regulatory subunit and the catalytic subunit such that the calcium channels can be opened. So this is one means by which the adenylate cyclase and the cyclic AMP might hypothetically be bringing about the opening of the extracellular opening of the ion channels in the membrane, so you get extracellular calcium coming in to trigger the exocytosis, and then a branching at this point, the 5B, for the pKa, as we would know, the dissociation of the regulatory and the catalytic subunits, and the catalytic subunit then can phosphorylate Krebs, bind to the CRE, and affect messenger RNA levels in affecting the receptor in this essence, as well as possibly, of course, the LH and FSH gene expressions. So this is a model for how the GnRH receptor might be affected. Well, I know that there are several other slides, so what I'm going to do is stop here and record the remaining slides in a separate lecture and have that posted. So I appreciate your time and attention, and hope to see you all again soon.